Ladies and gentlemen, please take your seats. Our program is about to begin.
Ladies and gentlemen, our program is about to begin. Please silence your cell phones or place them on vibrate.
أيها السيدات والسادة، سيبدأ البرنامج بعد قليل، لذا نرجو منكم التكرم بالجلوس في مقاعدكم.
It's day 9 of WEP.
We were always curious about the fish world and how can we change actually the ingredients and inspiration. Our main goal always was to see the sea and to see the ocean from another perspective.
I think a lot of us, even some of us that didn't realize how resilient they were, I think they discovered their own resilience through the last 2 years.
We'll see you tomorrow.
Assalamu alaikum. It's my pleasure to be here today. I'm Imad Gallouzi, Associate Director of Smart Health Initiative here at KAUST. I'm today on behalf of Pierre Magistrati, Director of Smart Health Initiative, who was supposed to be here, but he cannot make it for health reason. So today it's always a pleasure and an honor and really excitement to introduce a colleague like Dr. Kafui Dzirasa. He is, as you will see, he is an accomplished scientist, an outstanding speaker, and has many achievements during his career. He is an MD PhD. He did his degree, all his training mainly at Duke University, and his expertise is in neuropsychiatry and neurobiology. So at the moment, he is associate professor with appointment in several departments, the Psychiatry and Behavior Science Department, Neurobiology, Biomedical Engineering, and Neurosurgery. He started his MD PhD in 2001. However, before that, Dr. Kafui was a chemical engineering with interest in computer. From 95 to 2000, that's what he did. Then suddenly he got the inspiration and switched to become a doctor and a scientist. So he started his MD PhD in 2001, when he completed his PhD in 2007, and his MD degree in 2009, all at Duke University. And while he's finishing his MD study, he began a postdoctoral training from 2007 to 2009. So you will see throughout my introduction, he can do in parallel several things together and really excel in all of them. So in 2009, he began a career as an assistant professor in the Department of Psychiatry and Behavior Science at Duke. And then in parallel, while he's doing that, he began his residency. And when he completed it in 2016. And he was promoted to associate professor in 2019 with tenure. So Dr. Kafui is an outstanding scientist with amazing accomplishment. So his interest as a scientist he's studying how changes in the brain produce neurological and mental illnesses and how these changes can dictate behavior and emotion. So from the beginning of his career as a PhD student, he stood out as an excellent achiever. In fact, for example, his graduate work was recognized by several awards. For example, he was awarded the Samgen Award for Most Outstanding Dissertation Thesis, the Ruth K. Board Biomedical Research Fellowship, etc. So as a faculty, he built it on that outstanding achievement and continued in that path. So he published several papers, seminal, some of them are seminal. I will cite one. In 2018, he published a Cell paper, and in fact, that's a continuation of a long-standing collaboration with our provost, Professor Larry Karen, and both of them teamed together and used artificial intelligence and machine learning to apply in the neurobiology and neuropsychiatry. In fact, in that paper, they reported they used machine learning to identify and predict the emergence of major depressive disorders. And that tool has the potential to use this machine learning algorithm and capability to predict what's coming in terms of behavior. So, and today, so because of this achievement and many papers and awards he got, recently, in fact, I mean, we're reading his CV, I could not tell you or cite all his awards. By all accounts, it's 45 so far. Only last year, he had major ones. For example, he became a Howard Hughes Investigator since last year, and this is the top recognition and award that somebody as a scientist can aspire for in the United States. He was also elected to the National Academy of Medicine, and he was the laureate of the Benjamin Franklin Next Generation Award. This is only last year. So then with no further ado, today he will be telling us about a recent work as well as his journey and the resilience that took to achieve the work that he will talk about it, and I'm sure he will highlight some of his achievements throughout his career. So Dr. Kafui, welcome.
It's a tremendous honor and pleasure to be here with you all, some old friends and some new ones as well. Today I want to talk to you a little bit about my story and my journey. And in some ways, the theme of this winter enrichment program, resilience, I couldn't have timed it or planned it any better with the work that I'll be sharing about. So whenever I talk to young trainees, I always ask them about their why, right? So what is it that motivates us to do what we do? When I was an early scientist, I got really interested in the field of brain-machine interface and the idea that you could make prosthetic devices that were directly controlled by the brain. It's why I went to medical school and why I took all of my engineering skills and ultimately pursued a career in biomedical engineering. But I didn't actually find my why until I was in medical school, rotating in the inpatient psychiatric unit. And that's when I discovered the severity and the profound impact that mental illness had on people's lives. But it wasn't until I went home and started talking to my mom about my experiences. She's one of 5 siblings. That was the point that I realized that 4 out of the 5 siblings carried a diagnosis of depression, schizophrenia, or bipolar disorder. And as I returned to my classes in graduate school and I started learning about psychiatric genetics, suddenly I was terrified that one day maybe me or my brothers or sisters or one of our children would ultimately develop these illnesses. And so I decided at that time to dedicate my career and skills in engineering to apply it to understanding human health in the range of psychiatric illness and figuring out how to develop tools and technologies to ultimately treat it. Now, as I studied more, it became pretty clear that I didn't have a lifetime to come up with cures for depression and bipolar disorder and schizophrenia. So I started to look and ask myself what was convergent about the illnesses. And as my training went on, I realized that it was stress. And the idea was, stress was there at the beginning of psychiatric illnesses. Stress made psychiatric illnesses worse. And even when people were doing well, stress made the psychiatric illnesses reoccur. So if we could ultimately come up with a treatment to increase resilience, we could help alleviate all of the future stress and depression and psychiatric illnesses that would occur. And this is the framework that we thought our way through. And the idea is whenever you have people who are ill, have mental illness, we'll call that the susceptible state. And what our treatments try to do is they try to move the brain from the susceptible state into the resilient state. And if you look, you can see that stress basically causes the psychiatric illness to occur. What I thought at the time was perhaps there was a way to identify those that were at risk for future depression or future psychiatric illness. And if you could stratify those individuals, one from the other, and figure out what risk is, then perhaps you can come up with a way of improving resilience for everybody. So the goal was simple. We were simply going to see if we can predict who is at risk for depression and then ultimately come up with the way of preventing it. What we do in my lab and what I was trained to do was record electrical activity from many sites in the brain at the same time. And we do this primarily in preclinical animal models with ultimately working to come up with treatments for humans as well. And there are two types of electrical signals that we measure. The first one, we put wires each the size of a piece of hair into the brain, and then we're recording what we call local field potentials. And you can just think about these as what you classically understand to be brain waves. So we're recording brain waves from many sites simultaneously in the mouse's brain, and then we can do analysis, standard analysis that we do in engineering. We can filter those brain waves across frequencies and quantify how much activity there is in each site in the brain across frequencies. We can also take advantage of engineering principles that say things that change together across time tend to lie within the same system, so we can quantify how much these two waves tend to change with regards to each other. And you can see the peaks lining up here. We call this coherence or synchrony. In other words, these two brain waves are synchronized, and that's an indication that they're communicating. We can also take advantage of tools like statistical forecasting and ask if information in one brain area is best coupled to future information in the other brain area, and then we infer that information is moving from area 1 to area 2. Conversely, we can see if information in the other area is best predicting the future area information in the primary area, and then we infer that information is moving in the opposite direction. So as you can quickly see, we're recording activity from many sites simultaneously. Each of those brain waves has information across different frequencies. And then finally, we're trying to figure out if information is moving from one wave brain area to another, what direction is it going at? And pretty soon, we were essentially drowning in data. We were recording tons of data, and we actually didn't know how to make sense of it. And that's when I reached out to a colleague, one who you all know very well, Larry Karen, and his graduate student at the time, David Carlson. And Larry was working in the field of artificial intelligence or machine learning, and I asked the question, how can we make sense of all this data that we're covering? So I'll show you the model that we came up with, and I'll walk you through it. This is our newest version of the model, and we call it discriminative cross-spectral factor analysis based on non-negative matrix factorization. And if you want to understand the model, the easiest way to describe this is to think about music. Right? So whenever we talk about brain waves, right, and you hear sound, sound is also waves of activity, right? When you're hearing sound, you're hearing activity at the order of about 10,000 cycles per second. Now, if you hear music, you're hearing sound that's all mixed together. So think of the LFPs, the local field potentials of the brain waves, as if you're hearing sound. Then what we do is we chunk the sound up into notes. Right? And those notes are the measures that I just told you about. It's brain activity in different frequencies or power. Do the brain waves synchronize or coherence, or the directionality that it's moving in? So you take the music, and we change the music into notes. Then we use our machine learning tools to ask how those notes essentially play together. How do they change together across time? And so we can chunk those notes into chords. And we can also figure out which instruments are playing the chords. Finally, we relate those notes or those chords that are being played to what the animal's doing or behavior, or in this case, the state that the animal's in. Is it resilient or is it susceptible? And then finally, we add a last piece onto this machine learning model that asks, how can we make sure that these signals that we find work on a new set of subjects? In other words, if we want some sort of biomarker or diagnostic, we want it to work on the new patient that comes in the emergency room. So if, for example, new instruments are playing the music or there's a new tempo, one still knows that it is the same song. So this is the machine learning algorithm that we came up with, and ultimately, there's a lot of math behind this. So this is how we apply, I'll show you on the next slide, how we apply the machine learning algorithm to the problem that we're trying to solve. What we're actually going to do is we're going to create a similar sort of stress paradigm in mice. So we're going to implant the mice in brain areas that have been related to stress-related behavior. Then we're going to take the mice, and we're going to put them through a stress paradigm. These are all going to be genetically identical animals, and we're going to put them through a stress paradigm where they're essentially beat up or bullied by a really big, really aggressive mouse for 10 days in a row. At the end of getting beat up, if you have about 100 of these normal mice, they show a wide range of resilience. About 40% of the animals are resilient to the stressor. So they still show totally normal behavior even though they've been beat up day after day. The other 60% show a phenotype in which their sleep is disrupted, they look anxious, they no longer like sugar, and so they show a phenotype that looks a lot like how we would model depression. And importantly, if you take these mice that show the phenotype and you give them something like Prozac or a newer antidepressant agent like ketamine, it makes these behaviors go away. So this has been used as a model of depression. So what we're going to do is essentially record all of their brain activity before stress, put the animals through the stress, and then at the end, figure out what brain signature was there before stress that figured out what the animals ultimately became, susceptible or resilient. And when we do this, we come up with these maps. I call them electrical functional connectomes. And these are our two first maps that we published in 2018 in a paper that Larry and I worked on together. And I'm going to orient you to these maps. The map on the right is what the depression signature looks like. So this is what the signature looks like in the animals' brains once they're showing the depression phenotype. The map on the left is what the animals actually look like before you stress any of them. So they're behaviorally the same, they're genetically the same, but their brain maps look very different. Around the rim of the circle, you can see each of the brain areas that we've recorded. So ventral tegmental area, BLA, or basolateral amygdala, infralimbic cortex, prelimbic cortex, nucleus accumbens, and ventral hippocampus. So the areas around the rim, the frequencies, in other words, the frequencies of the waves from 1 to 55 hertz are around the rim of the circle for each of the brain area. If there's color on, let's say, the tire part of the wheel, it means that brain area in that frequency is part of the network. If you see a line, essentially spokes, connecting the two parts of the circle, it means those brain areas synchronize. And on the bottom, if the brain area is synchronized, you can essentially see how information is flowing through the circuit. So we've color-coded this so you can see the brain networks look very different in the depression phenotype, which is on the right, and the vulnerability or the prediction signature, which is on the left. So we're really excited about this. What this meant is now we can look at mice that all behaved the same before they were stressed and find a resilient signature in the brain, right? So we can find the risk in the brain. So, I'm about 7 or 8 minutes in, right? I said my goal was to find risk prediction and prevention. So clearly, discovering prevention was not as easy as finding prediction in the animal's brain. So I'll spend the rest of the talk talking about how we ultimately got to prevention. And there are two things that I want you to pay attention to as I'm giving the talk. One is the science, but secondly is the story of resilience behind the science and the journey that we took. And I'll give you a heads up. As we're doing this, being resilient actually is going to require us to course through a series of scientific disciplines, many of which we never thought about before we started this journey. So I'll orient you to each of the scientific disciplines, and then I'll show you how we sort of brought ourselves into this space in order to solve a problem. And for me, that problem was ultimately figuring out how to come up with new treatments to help my loved ones, my family members. So we started off just thinking about simple stress paradigms that almost everybody uses, right? So this is a classic stress paradigm utilized in mice. It is not depression, but what it certainly does is it induces a stress response in mice. So if you look at the mice, it makes their stress hormones levels go really high. So that's one feature of the test that's really important. The second one is the test, the assay itself, is animals behave differently if they've been previously stressed. So if an animal's been stressed and you put it in this situation, either putting it in a tank of water that it can't escape or hanging it upside by its tail, animals that have been previously stressed stop trying to escape. They essentially give up, right? So the test is stressful, and the animals that have been stressed try to give up. It creates a really nice feature in this test, and that is that you can then put the animal, if you put the animals multiple series in a row, the animal actually changes in that the first test is stressful, so the second time it gives up much quicker. So the stress causes the animal's brain to change such that it adapts to the stressor and its behavior changes. So it's a really nice test that we can use to study how the brain changes in response to stress, giving us an opportunity to find resilience. Here's what it looks like. These are, on the left, you see animals that have been put on the test, in other words, hung upside by their tail on day 1 and day 2. The y-axis on the left tells you how much the animals move, and you can see as a group or population of animals, they move less on the second day. In other words, they get more mobile. So the stressor causes their brain to adapt. And importantly, this is context-specific. So if you put the animals in another situation, you just let them run around the cage for food, the amount that they move in that arena doesn't change. So their brain only changes, or the computations change, in the stressful condition. So what we're going to try to do here, it's another set of machine learning tools that we applied. We just basically record activity from all the brain areas. We're recording brain waves, but now we're also recording single cells in the brain generating electrical pulses, right? You have about 100 billion cells in the brain that can generate these electrical pulses. And then what we're going to do with Larry's group is ask the question, how do the electrical pulses in the brain change? And are there specific brain regions in the brain that are responsible for the animal's behavior changing between day 1 and day 2? In other words, how is stress changing the computation in the mouse's brain? And when we do this, out of all the brain areas we record, we actually find it's only 2 brain regions that are changing, right? It is one in your frontal cortex called infralimbic cortex, and another one in a region called thalamus. And I'm going to put placeholders there. I just want you to hold on to that. Don't get too deep into the details, but it's only 2 out of all of these brain regions. So then what we're able to do is zoom in on the activity of these brain regions to learn how they change in response to stress. Because if we can learn how they change in response to stress, we can come up with treatments that prevent stress or play resilience into the brain. We then zoom in, we take the brain waves from each of these brain areas, and we put them through the same series of analysis that I showed you originally, right? Is the activity different in different frequencies? Do they communicate in a different way? And the sad answer was, no, the broad activity, the brain waves in each of these areas was not different between day 1 and day 2. If we looked how these areas were talking to each other, there was no difference on day 1 and day 2. They were communicating, and their communication was going in the same direction. But then we asked a slightly different question about how they talk to each other. And instead of asking about how the waves lined up, we actually asked if they were communicating across different frequencies. And the best way to understand what I'm saying here is to, again, think about music, right? And so you can have activity of different frequencies that synchronize, almost like, right? So you have different frequencies that are synchronizing together, and we asked that question, right? Are they coupling across different frequencies? And here you can see a low-frequency wave in green, high-frequency wave in red, and you can see that the peaks, or the amplitude of the high frequencies, is coupled to the lower frequency. And when we ask that question, here's a plot of coupling between different frequencies. So the lower frequency brain area is on the bottom, from 0.5 to 9.5 cycles per second. The higher frequency is on the y-axis, 15 up to 70. And the color there signifies that the 2 areas are talking across frequencies. So we found that these areas were talking across frequencies, and that communication was changing between day 1 and day 2. So we found a signature that was related to the animal's behavior changing in response to stress. So we were really excited about this until it occurred to us that we thought a lot about physiology in the classes that we'd taken in medical school. And when you have a physiological signature, that physiological signature can easily be one of 2 things. First of all, it can be primary. In other words, that signature is what's causing the problem. And so what that means is if you can get rid of the signature, you get rid of the problem. Alternatively, it could be compensatory. If that signature is compensatory, it means it's the body's way of trying to counterbalance the problem. Think about it. You start, everybody, if we all to get up and run outside, our hearts would start beating faster. And the reason our heart is beating faster is because it's actually trying to deliver more oxygen to our muscles, right? So the heart beating faster is a compensatory response. So the problem is, if this is primary and you get rid of it, the animals are more resilient. If it's compensatory and you get rid of it, the animals are worse, right? And certainly one thing we don't want to do is we're trying to make people resilient is to actually do the opposite thing, get rid of compensation and make it worse. So this really matters. And this was the first problem that we ran into. Because in order to figure out if animals were having a primary signature or compensatory, we would have to recreate it. We'd have to some way figure out how to play this signature in the brain. And figuring out how to play this cross-frequency pattern into the brain weren't tools that really existed. And so we shifted over to biomedical engineering, and we thought of ways to create it. And to create this, what you ultimately want to do is come up with a tool. We call this NCLASP. And what this tool essentially needed to do was to read brain waves in part of the brain and then play the right wave into another part of the brain. So the two areas that we found were changing, we had to read one and then play that brain activity into a second part of the brain. And what made this really challenging is these brain areas were talking to each other with a delay of 30 milliseconds, right? So 1/30th of a second. So we had to extract this information, process it in a computer, figure out where in the wave the brain was, and then play the higher frequency activity in a second brain area. But we came up with a tool to do this. And you'll see here, essentially, we extract the information, process it in a computer. We have to do this in about 30 milliseconds, and then stimulate activity back in the brain. And here, I'm going to orient you. You're going to see blue and yellow. We're going to use a tool here, which is a protein. It's the same sort of protein that's in your eye that allows you to see light, rhodopsin. And that channel basically changes light into brain energy. And so the channel that we're going to use in this case, it is activated by blue light. So blue light is going to turn on. It's going to be our stimulator. Yellow light is going to do nothing because this channel doesn't respond to yellow light. So we show we can do this quite accurately. So we can extract the waves in black. You see the waves. And then the blue is a stimulation. So we can generate that high-frequency signature on top of the low-frequency wave. We can do this quite reliably. And then all we're going to do experimentally is we're going to put an animal in the stress condition and play the signature in its brain. And I'll remind you again, if it's primary and we play the signature into the animal's brain, the animal should get worse. And if it's compensatory, and we play the signature in the animal's brains, the animal should get better. In other words, they should move much more in this stress condition and continue to try to escape and be resilient. So we did this experiment. We were really excited. We built this in the lab. We put animals through this paradigm where we're playing blue and yellow. Blue again, we're playing the signature in the animal's brain. And you can see all the way to the right, I'm showing you immobility here. The blue animals are less immobile. In other words, they move much more. And so what this immediately suggested to us was that this was a compensatory signal. We were actually playing a compensatory signal in the animal's brain, and back into 2014, we had succeeded in figuring out how to play resilience into the brain. And we did two important controls, just to make sure we hadn't lost it here. The first important control we did is we got rid of this sort of what we call closed-loop stimulator, in other words, reading and writing the brain, and we said, could we just activate these cells without the need for all this complex engineering and complex math, and let's see what happens. So we put the exact same number of activity signals into the animal's brain. You see that on the left. And when we put the exact same number of activity signals without making sure that it was with the right timing, the animals actually got worse. Right? So the animals became more mobile. In other words, we turned off the compensation all the way and made animals worse. And then alternatively, if we just put the 3 pulses in the brain, in other words, the pattern was the same, but you can see when we did this, sometimes you would line up with the right part of the wave, sometimes with the wrong one. It had no effect at all. So the only way to get the animals better was to essentially create an engineered system in which we're reading from part of the brain, writing in part of the brain with a 30 millisecond delay. So I'll just sum this up. When we put in the light pulses and they were timed to the brainwave in the right place, the animals got better. When we just stimulated the brain without patterning, so continuously, this is what most of our deep brain stimulators do when we're treating when we're trying to treat mental illness or Parkinson's disease, the animals actually got worse. And if we just put in 3 pulses, you can see sometimes the pulses end up at the bottom of the wave, sometimes at the top. They all cancel themselves out, and nothing happened. So we figured out here that we had a very viable pathway to ultimately improving resilience. All we needed to do was make closed-loop stimulation devices that could be implanted in the brain, and we would ultimately have our signal to creating resilience in the world. And this is what it would look like. And we popped a bottle of champagne, we published this paper with Larry, and we're really excited about this. And before you know it, I ended up with my family. So this is my family, this is my why. And this was at my grandmother's funeral. My family's from Ghana in West Africa. And there's an open chair. And the open chair is my uncle, so my mother's brother. And at my grandmother's funeral, he wasn't there because the stress had led to a reoccurrence of his bipolar disorder. And this was a really monumental occasion for me because I remember talking to my uncle and having a conversation about how we created this new tools that were going to increase resilience and help my family members. And I had a conversation with my uncle that never left me. And he said, we're really proud of you. We feel like it is our family's fate that we've suffered so that you've gotten motivated enough to create something that might ultimately help the world. But we're proud of you, but I really want to understand, so these microchips that you're creating, that you're going to implant, So tell me more about how they're going to get to the third world. And it occurred to me as I was sitting there with my uncle that I'd spent my career working on a technology that was unlikely to help my family because it was never going to get to the third world. And as I sat there, devastated in this moment of triumph and success, I was immediately devastated. And it forced me to take a step back and ask the question, what sort of treatments actually make it to the third world? When we're talking about illnesses like depression or bipolar disorder or schizophrenia or mental illnesses, they yield the greatest burden of illness worldwide. And so if we're going to move that needle in any way whatsoever, we need to be thinking about creating tools that will ultimately make it to entire parts of the world. And certainly this was before COVID-19, where we all realized how hard it is even to get vaccines to the rest of the world, the third world and the developing world. And so at that point in time, I had two revelations. The first one was, in order to make headway, we need to do one of two things with the technology that we're creating. We either needed to turn it into a pharmacological agent, in other words, pills, because people know how to get pills to the third world, or we needed to turn it into a vaccine. And this seemed impossible, right? I mean, we've essentially created this in silico, on-chip device, and somehow I needed to turn it into a pill or a vaccine. And simultaneously, the US had launched what they called the Brain Initiative, and the idea was to understand the human brain. And there were two technologies that were being developed that convinced me that perhaps we had an opportunity of turning this device into a vaccine. The first one is, it was working on sequencing all the cells in the human brain, essentially coming up with a GPS code, the gene expression patterns for each cell type. Simultaneously, other investigators were working on coming up with AAV viruses, in other words, viruses that cross the blood-brain barrier. And the idea is if you can connect these two, you can deliver protein packages to specific cell types in the brain. So if you could turn that closed-loop stimulation device into proteins, there's a way you can ultimately turn it into a vaccine packaged product. So our goal was simple. Take this closed-loop stimulator and turn it into proteins. Now, obviously, this was daunting. As we started talking about this, we knew it was unrealistic. We often heard that it was an impossible feat, but this is what was necessary to ultimately help my family. And as you all know, we are all motivated in tremendous ways when it comes to our loved ones. So here's what we're going to try to do. We're going to try to create a protein-based tool that does the following. You've got to be able to target it to specific brain cells. So it's got to go to specific circuits, right? Again, I showed you that only two brain areas change, so we don't want it to go everywhere. We want it to go those two brain areas. Secondly, it can't require external stimulation, right? So it's not going to be battery-powered. It's not going to be a device. It's got to use brain energy to do the closed-loop stimulation. Thirdly, our closed-loop stimulator read from a brain area and played activity in another one, so it's got to have a preferential direction. And then finally, we've got to make sure that when we put it in, it doesn't ruin everything else that the brain is doing, right? The last thing we want to do is cause problems. So this is ultimately what we're going to try to solve. And we wrestled with this. How to do this, we pulled together what we call our thought team, and these are generally tend to be younger scientists, sometimes undergrads, who think more creatively and outside of the box to try to solve this problem. And ultimately, we realized that this problem had actually been solved by nature. So as you all can appreciate, this is the heart. And the way that the heart works is it's able to synchronize itself, which is why it beats. And the heart is able to do this based on a set of proteins called gap junctions. And the way gap junctions work is that they're essentially pores that let ions flow through them. So instead of the classic chemical synapses we think about, these are electrical synapses. And what's so important about these electrical synapses is that they basically take electrical signals from one cell and pass it to the next cell. In other words, they work as closed-loop stimulators. They read and they write, and they're powered by biological electricity in the same way that your heart has waves that are synchronized across the heart. And so we thought to ourselves, perhaps we can take these proteins called connexin proteins. The connexin proteins bind together into 6-part channels called hemichannels, and the hemichannels come together to form a gap junction. Perhaps we can turn these into our closed-loop brain stimulators. Now, there's some problems. If you do this with connexin proteins, I'm showing you here a nervous system. Imagine 2 cells coming down, and those neurons connect to another 2 cells on the bottom. If we were to put in gap junctions, or these hemichannels, on the left hand, you get what you want, right? We can connect these 2 cells together and let electricity specifically flow through them. In other words, change the dynamics of the neural circuit. The problem is that these Hammy channels bind to themselves. They're like stickers, right? So we would immediately cause other things that aren't connected in the brain or don't have strong connections, we would put the connections there too. So you can see the two cells on top, we would also put connections there. So while we can target it to specific cell types and it doesn't require external stimulation, it doesn't achieve the other two things. And if it doesn't achieve the other two things, you can cause the brain to become inactivated in the wrong way. In other words, it's not preferential, and it can mess up the rest of biology. So what we're going to try to do is solve this in a slightly different way. And what we need to do is take these connexin proteins, these proteins similar to the ones that are in the heart and the rest of the body, and we need to change them. We need to change them so instead of working like stickers that stick to themselves, we need to engineer them so they work like magnets, right? So we can have a positive end and a negative end. And if we engineer them to work with like magnets, then the positive will repel the positive, the negative will repel the negative, but the positive and negative will come together to essentially form a channel. And then we can send each a half of that channel to where we want them to go in the system. So now, all of a sudden, we started off with a neuroscience problem, then a machine learning problem, and then a human problem, and all of a sudden now we have a biochemistry problem. And having almost failed biochemistry in medical school, we were certainly way outside of my wheelhouse. And so again, we pulled our undergrad team together, and they basically went through all of the Connexin literature. Again, I love having undergrads and young scientists work on projects because they think creatively and they think way outside of the bounds that science, our training has forced to think us within, has taught us to think within certain boundaries. And our team of undergrads went through all the literature. They looked at connexin proteins, which are the mammalian version. They looked at innexin proteins, which are in invertebrates. They looked at panexins. They looked through all these different proteins that form gap junctions and actually settled on these two. And they settled on these two. They're in fish, perch fish, and goldfish, and the connexin 34.7 and connexin 35, and they chose them because these connexin proteins do something that's really cool. They send current in a preferential direction. Right? So most connexin proteins are just like pores. These connexin proteins actually send current in a preferential direction, and these two different connexin proteins can bind with each other. So now we have connexin protein A and B that bind with each other and send information in a preferential direction, so we decided to start with these. The problem with them is that while 34.7 are A and 35 bind with each other, they also bind with themselves, right? So they're like stickers that stick to each other. So what we're essentially trying to do and what we're trying to achieve is a very complicated yet intellectually straightforward biochemistry problem. We have to engineer the part where they connect, and this is known as part of the extracellular loop. In other words, the part of the connexin that's hanging outside of the cell. We just have to engineer the sequences, the pluses and the minuses of the amino acids, and change them from stickers to magnets. Now, I describe this as I'm saying this. It seems pretty simple and straightforward. It's never been observed before in nature, and we didn't have a great background in biochemistry to know how to do it. And so at that point in time, I recruited a very ambitious graduate student that I've been mentoring for 3 or 4 years to the lab to start working on this, Liz Ramsey, who'd been finishing up her PhD in chemical biology at Harvard. And the way one would classically do this is you'd set up a system and maybe 2 neurons, and you would hook them up so you can record electrical activity, and then you put one connexin in one connexin in another, send electrical signal, and then measure how well the electrical signal is moving. The problem with taking this approach is that it's slow, right? So you can screen one connexin at a time or one pair of connexins at a time. And we realized that in order to solve this problem, create something that's never been observed in nature before, we're going to have to engineer an entirely new way to rapidly screen lots of connexins over and over and over again. And so before you know it, now we're suddenly in a new discipline in a new space again, right? And I think part of the lesson I'm trying to convey in this story is resilience requires you to constantly adapt to a changing environment to solve problems. And so we're going to go back to that light-based tool I mentioned, channel rhodopsin, or the rhodopsin, same protein that's in your eye, that takes light and turns it into energy, right? So we can take light now and turn it into electricity. And then we're going to borrow another tool from a colleague at MIT, Ed Boyden, that takes energy and turns it into light, right? So one turns blue light into energy. The other one takes energy and turns it into red light. And what we can do now is set up a system where perhaps we can change blue light into energy, energy into red light, and use that as a way of measuring current flow. And the system is going to be pretty simple, right? Instead of using neurons, we can use cells that multiply and divide pretty rapidly. These are HEK cells, or human embryonic kidney cells. And then we're just going to engineer them to act like neurons, right? So we're going to put proteins in one, the cell on the left, and this is a potassium and sodium channel, so that it sort of creates an action potential like a neuron. And then we're going to put the rhodopsin, or the channel rhodopsin, in it. So in other words, if you shine blue light, it makes that cell create an electrical pulse. And then on the second cell, we're going to put that voltage indicator, which is going to take electricity and turn it into red light. And so if current can move between the two cells, you have a system now that changes blue light into red light. If current can't move, you put in blue light, you don't get red light out. And then we can put a connexin protein, each of our different engineered connexin proteins, one into each cell, and then just test which cells change blue light into red light. And now, like these screening systems that many universities have, you can put a connexin protein in each well plate, shine blue light, see if it turns into red light, and instead of moving one at a time, we can move thousands at a time. Right? So we took the time to try to set up this new system so we can really quickly go and find our master pair of connexin proteins that we're actually going to use to create this new tool. And I'm just showing you here, these are images. In the green, you see the sensor, or the voltage indicator. In the red, you see the channel rhodopsin, or the blue light exchanger. And we were able to create cells that had all of the necessary parts to create this system. It took about a year, but we were really excited about this. And then we started putting our connexin proteins in to test the system, and every single cell died, one after another. And we couldn't figure out why the cells died. Ultimately, we think we were putting in too many things into these HEK cells that required energy, and they just didn't have enough mitochondria to keep up, and so the cells died. But after this entire year of planning, of creating this entirely awesome experiment, all of the cells died, right? And so suddenly we didn't even have a system to test these connexin proteins that we were creating, which was this really hard project. And I'll show you this image. Liz was looking and she was watching over and over these cells die, which is a really hard thing for a postdoc to see occur in their career. But she kept looking at them over and over and over again, trying to figure out what was going on. And as she was observing these cells, she kept noticing these like little small bubbles inside of the cell. And here, our connexin proteins are tagged with fluorescence, or in green. So you can see the green bubbles inside the blue, which is the cells. And she just saw these bubbles over and over again. And so then she started going back and reading more about gap junctions and connexins and how they operate in cells. Again, none of us have a background in any of this stuff. We're just trying to solve a problem. And when she did this, she realized that there's a life cycle. In other words, how connexins live and thrive in cells. And here's how it works, right? The connexins are basically synthesized, and then they're moved up. They form the 6 channels, and then they're moved 6 protein channels, and then the hemichannels are moved up to the surface of the cell. That's where they meet the hemichannels on another cell and form a gap junction. Now, when you want to get rid of these gap junctions or downregulate it, you actually internalize the whole plaque, right? So you basically absorb the whole gap junction into one cell. And it occurred to her that she could actually use this life cycle as a way of screening whether connexin proteins were coming together or docking. So what she did is she added different colors to the connexin proteins on different cells. She put red on one and green on the other. Sorry if you're colorblind. And her idea was that if these connexin proteins came together, they would essentially be sucked into a single cell. So you can tell if you formed a gap junction, because if you have green and red in the same cell, you succeeded in forming a gap junction. And so when she did this, you'll just see the movie play. She's plated green cells and red cells. You can start to see in the upper left-hand corner a bubble with green and red in it. And so we were really excited when we started seeing individual cells with green and red in it, because it meant that we were now have a way of screening whether our connexin proteins were coming together. And so she then took these cells, using this method that she'd created, and went to a flow cytometry machine. Again, this is a new experience for me. I'd never used flow cytometry before. Apparently, a lot of our colleagues in cancer research use flow cytometry all the time, or FACS sorting. And what this essentially does is it separates individual cells in a population of cells and tells you how much of each color is in each cell. And so this is what it looks like. She basically takes some of our connexin proteins that are tagged with the green, some of the connexin proteins in cells that are tagged with the red, and puts them together. Then lets them incubate. and then takes it to flow cytometry. And in the flow cytometry, you basically get a readout, and the readout you see on the bottom right. On the y-axis, you see how much green there is. On the red axis, you see how much red there is, and each dot is one cell. So on the bottom, you could see a sample in which cells only have green or cells only have red. And on the top, you see some that have sort of moved to the upper right-hand quadrant. These are cells with green and red. So she can quantify out of our population what percentage of the cells have green and red. So if the two proteins don't come together, you get the plot on the bottom. If the proteins come together, you get the plot on the top. And so suddenly she had an entirely new screening system for all the connexin proteins we were going to engineer, and we can now rapidly go through them, right? We'd essentially done what is we call going slow to go fast, right? We do all of the slow engineering so that we can move really, really fast with the screening. So when she did that, and she decided to take it through a three-step approach. So first, we were going to take each connexin protein and each mutation, and we were going to see if it docked with itself. So we want to identify those that don't dock with themselves because we've randomly mutated a bunch. Then we want to test those that don't dock with themselves and see if those combinations dock with each other, right? So doesn't dock with itself, docks with the opposite one, and then any pairs which survive that process, we were going to test whether those parts dock with any of the other connexins in the human body, right? Because what we don't want to do is create really bad connections that we don't want because they're connexin proteins in the human brain, and our goal was ultimately get to humans. So we want to make sure a connexin protein doesn't dock with itself, it docks with the other part, and then finally it doesn't dock in the human body. And amazingly, we went through all this process. We started over with 140 candidates. 15 candidates made it through the first round. 3 pairs actually made it through the second round, so we're really excited about this. And when we compared those 3 pairs against all of the other connexin proteins, the major connexin proteins in the human brain, we found that they were actually docking with connexin proteins in the human brain. So again, this entire project fell apart. We had learned something, we'd made tremendous progress, we'd engineered a new feature in nature that had never been observed, but it wasn't going to meet our goal of actually putting this in the human brain in the long run. And then things got so much worse. That result came in the spring of 2020. And suddenly, when we made progress and we felt like perhaps there was some level of hope to this, the entire lab shut down. And like most people in, like the folks in my lab, I'm sure you all will also share the experience of making major changes in your scientific career. We all became bakers and cooks, right? Everyone started baking French pastries. And so we all stopped doing science and shifted to a new range of science. But interestingly enough, I had a student in my lab whose background was computer science. And instead of baking, he actually convinced me and the postdoc to buy him a supercomputer, which he then installed in his house. And what he decided he was going to do with the supercomputer was he was actually going to use machine learning to start looking at protein structures. And so he went through the literature and found protein structures for connexin proteins that have been solved, crystal structures, and went through and started creating a mathematical or a machine learning system that would actually create models of connexin proteins. And so then he could take all the mutants that we created, he could feed them into the computer. We knew the ones that disrupted docking. We knew the ones that made them dock with each other. He fed everything into a computer, and he actually learned how these proteins were interacting with each other, the amino acid residues, in other words, the sites on the extracellular loop that controlled how connexin proteins were docking. And suddenly, he had a model, right? A model out of machine learning that suggested the important interactions between these two proteins that determined whether these connexin proteins would operate like stickers or magnets. And he was able to find this pattern that you see on the right of where the attractions and the repulsions were. Taking this information, in silico, he started engineering new proteins. So he decided to basically start off and basically change one at all the key residues to positive. On the other protein, he changed it all to negative. And he said, well, this should solve our problem, right? We should essentially turn these stickers into magnets. The positives will repel the positives, the negatives will repel the negatives, but they will dock with each other. And so about 4 months later, the lab opened up again. He now had this computational model, and so we created these connexin proteins. And when we created these connexin proteins, we created them, we expressed them back in our cellular system, and the one on the right, you see that the connexin protein expresses just fine. It makes it on the surface of the cell. The one on the left didn't work. Right? The connexin proteins actually got stuck in the cytoplasm. So this was a really great example for us, right? We spent a lot of time doing machine learning, right? This is why validation is important, right? Because solving it on a computer doesn't actually mean it generalizes to the real life situation, because the computer analysis that we created never considered whether or not it would traffic to the surface of the cell. It said if it's trafficked to the surface of the cell, it will dock. But we made some slight modifications given all the information we had. We just changed one of the residues on here. So we created a slight repulsion, but not a huge one, and went back, and all of a sudden the connexin protein made it to the surface of the cell. So by combining machine learning and protein modeling, right, creating this screening system and engineering these proteins, we suddenly had two proteins which only docked with each other, but didn't dock with themselves. And when we went through and screened these proteins against the connexin proteins in the human brain, they didn't dock with those either. Right? So suddenly we had our candidate protein pair, right? Our two proteins that we can use to essentially create a closed loop system. All right, so at this point in time, what most scientists would do is now go back to the slow system, patching the two cells, put each of these sort of halves of the protein closed loop simulator in the two cells, and see if it works. But I am an extremely impatient person, and almost every day I'm driven by the idea that I'm going to help my family members. They're not getting any younger. And so we decided to do something way more aggressive. We reached out to a colleague of mine who was working in an entirely different area of neuroscience. And he was working in an entirely different model organism, in this case, a worm, or the C. elegans. And for me, I will be honest, during most of my career, I never really thought the worm was going to be that useful for how I think about mental illness, right? I'd never thought about the worm as useful for thinking about resilience. But there are a couple of important features that this worm has. One, it has about 302 neurons, exactly 302 neurons, that have been totally mapped out. The wiring diagram is totally created, right? And then the second thing that's important about the worm is they're see-through, so they're translucent. And many of the behaviors that the worm engages in has been mapped directly on this circuitry. So I reached out to a colleague. An important part of resilience is the community around you. So I reached out to my colleague, and I'll show you what he did. So he'd been studying a circuit between two neurons, and one of those is a sensory neuron. It's called AFD, and the other is an interneuron called AIY. And when you feed these worms at cold temperatures, the worms search for food in cold temperatures. So if you feed the worm in cold temperatures and you give it a choice, it goes to cold temperatures. And this choice of going to cold temperatures is controlled by these two neurons, right? So at least in part. So the first one is the sensory neuron. So you can see the plot directly under the sensory neuron. As you raise temperature, the activity in that sensory neuron goes up. This is calcium imaging of the cell. It's a proxy of activity of the cell. So temperature goes up, activity of the cell goes up. That cell sends information to the interneuron, and if you fed the animal at cold temperatures, as the sensory neuron goes up, the interneuron doesn't change, right? So sensory neuron increases activity, but the interneuron doesn't change. It doesn't talk to it. But if you feed the animal at warm temperatures, and again, the animal prefers cold temperatures, if you feed the animal at warm temperatures, these two neurons now talk to each other, right? So as temperature goes up in the sensory neuron, you see the calcium rise. If you feed it at warm temperatures, the activity also goes up in the second neuron. In other words, they now send information to each other, and then the animal performs warm temperatures. So what we thought we could do is use this as a screening system to determine the connection between two neurons. And the idea is, if we just stick a connection between these two neurons, we can change the animal's behavior. So we started off with these two neurons, and we put connexin protein A into both neurons, right? Again, connexin protein A should repel itself. So we put connexin protein into both neurons, and then we let the animal explore a temperature gradient, right? When we do this, the animal, the neuron, the sensory neuron, responds to temperature, but the interneuron doesn't. So in other words, if you put half, the positive side of the magnet and the positive magnet in both, or the negative side of the magnet and the negative side of the magnet in both. There's no communication between these two neurons, and the animal, when it has a choice, goes to the colder temperatures. However, if you put our two proteins, right, our engineered proteins, the positive and the negative end, between these two channels, now all of a sudden, when you increase temperature, the sensory neuron goes up and the other one goes up as well. So now you've basically engineered an electrical connection between these two cells using our two proteins. And here's what the animals look like behaviorally. You see that on the right. Now, on the right, you can see that now, by putting our protein in, the animal's brain changes. In other words, these two cells talk to each other. And when you look at normal animals, they prefer the cold. If you put 34.7 and 34.7, in other words, the positive magnet and the positive magnet in, the animals prefer the cold. And if you put the negative magnet in and the negative magnet in, the animals prefer the cold, right? It's only when you put the positive and the negative in between these two cells that suddenly the animals prefer the warm. In other words, you have created a closed-loop electrical pacemaker that is proteins between these two cells. All right, so suddenly, we'd gone slow. We'd been working on this for years and years and years and years, and last summer, we found that we'd solved this problem in worms, right? But it is the case that none of us are worms, our family members aren't worms, and so we wanted to ask the question, could we actually use this in the mammalian brain? And so we went all the way back to the beginning again. We went back to that original problem we were trying to solve about how mice change in response to this stressful environment. And what we were going to do is quite simple. We knew the two brain areas that talked, and we knew exactly where we deployed our closed-loop brain stimulation system. And I'll remind you again, we knew how the two brain areas talk to each other. We'd already gone through and created this closed-loop stimulation system and shown that this was a compensatory circuit. And this compensatory circuit, when it operated right, in silico, right, on chips, we can make the animal more resilient to stress. In other words, we can play resilience onto its brain. And so the question became, can our proteins do the exact same thing that this in silico chip was doing? So our experiment was quite simple. We put one side of the magnet, in other words, one half of the connexin protein, in one of the brain areas that was important, the one we were reading from. We put the other half of the connexin protein in the second brain area, the one we were writing to, and then we just let them express in the animal. And ultimately, what we expected it to do was just to form an electrical connection between these two cells that was one direction. In other words, a closed-loop brain stimulation system made out of proteins. And one of them was tagged with a green fluorescent protein, one of them was tagged with a red fluorescent protein. You could see the images on the right. And then we just put the animals through the exact same assay. And we're going to test these animals again. We're going to put them on the assay on day 1, and we're going to put them on the assay on the second day. And our hypothesis is, if we've recreated our closed-loop stimulation system, the animals should be more resilient to stress. And I'll never forget, I was flying on a plane. I had internet, flying on a plane from California, and the senior scientist in my lab sent me this figure. And what you will see on the left hand, this is how much the animals move in this stressful condition. The animals in blue are the ones with either the positive or the negative. In other words, they are control animals. And between day 1 and day 2, they become more mobile. In other words, they show the normal stress response. But in the animals that we put the lock and the key, they did not change at all between days. In other words, we had programmed resilience into their brain using proteins. And when we looked at these animals in an open field, in other words, running around and looking for food, there was no changes at all. We had specifically hit the circuit that turned on resilience in these animals' brain. So I don't know where this is going. This data is about 2 months old. We're now trying to work to see if we can target other circuits and thinking about ways that we can put this into human brain cells as well. I know one thing for sure. We're substantially closer than we were 4 or 5 years ago to coming up with a treatment that might one day help my family. And I think this has been a real lesson for me about the need to take risks and to cross boundaries and how much failure goes along with making substantial progress in science. I'll leave you with this last slide. I think it's important because it represents the people that did this work. And I always say, with respect to everyone in the world, this is the power of U.S. science. And it's the power of bringing together people from all kinds of disciplines and all kinds of backgrounds, right? Some of the people here are first-generation college students. People have migrated from other countries. There are people who are young parents. I think for me, we would have never solved this problem without so many people that think differently. And I hope that next time I give this talk, there'll be a couple of folks who trained here, KAUST, who are in the lab or from other parts of the world also, bringing your expertise to solving these problems that really matter for improving human health and the human condition. So thank you so much for having me here. It's been a real honor and a pleasure.
Thank you so much. Very inspiring. Wow. So we have time for a few questions from the audience. We have 2 microphones on each side. Please, if you have a question, come to the microphone. Arnhem? Yes.
Very inspiring talk indeed. Really enjoyed. I have a question related to the gut-brain axis, and I'm wondering if the stress could be also, made resilient through some sort of, manipulation of the gut microflora, and if that that could also be part of a future treatment strategy.
So yes, and let me say I'm a neuroscientist, right? Which means we have an obsession with the brain. And that unique obsession means that sometimes we think that the way the brain works is solely captured by your head within your cranium, and it is not, right? You certainly have 2 synapses from the heart into the brain, 2 synapses from the gut to the brain. The immune system impacts the brain as well. So as a physician, I think a lot about how the brain is actually integrated with the body as an entire continuous system. There's no doubt that there are more cells in your body that are gut bacteria than cells that you would otherwise consider you, and they synthesize proteins and other factors that impact brain function as well. So yes, I think targeting the gut-brain axis is going to be really important, both in terms of targeting the microflora, but then also having tools that can target the neural information and the connections between gut and the brain. One of the reasons we like this new tool so much is because it's actually not just limited to neural interactions in the brain, right? Because the stimulator essentially floats around with the end of the nerves. And so the stimulator itself could be applied at the gut-brain axis or between interfaces between the brain and other parts of the body as well.
Thank you.
Yeah.
Question?
Hello. I'm Salim Maldajani. Very, very inspiring talk. Thank you for sharing, all of the successes, but also all of the challenges that you faced along the way. I had a lot of questions, actually. I submitted them all virtually, but I chose the one to ask here in person based on the upvotes that I got. So, this type of research is pretty tricky because of all of the regulations involved and the ethics involved. So how do you deal with the ethics involved with the stress experiments? Like, for example, what happens if a mouse drowns? Are there clauses in the proposals that protect the medical subjects? And how do you prevent, Stanford prison experiment type outcomes in these experiments that, can have negative outcomes?
Yeah, so let me say 2 things. One which I'll say in sort of a humorous way, but then answer quite seriously. Growing up, there were mice in my house, and my mother never thought about the neuroethics of the mice, right? And so I think about animals as being quite distinct from humans. That is my joking answer, but yet quite seriously. I think there's a massive difference between the suffering that we see of human beings and how we think about animals. Nevertheless, it is very important to have animal parameters in as well, right? We have IOCO committees to take care of the animals. But I think it's ethical to do stress experiments in mice to ultimately cure depression in humans, partially because my mother never thought about the mouse trap being unethical. So we do put things in place, like we worry about the mice, and so we watch them as they're swimming, and so we don't let them drown. because we're standing there watching them, right? As we're putting them through the stress paradigms where they're essentially getting bullied, we make sure that they're never having serious injury, or we remove them from the task, right? So those are the ethical things that we think about with regards to the mice, but the ethical standard that we have for humans is way higher, right? We don't put humans in the water tank, right? In an ideal world, we would figure out how to remove the stressors that the humans would experience that would cause the pain or the trauma, right? But we certainly know our police officers or firefighters are going to experience some degree of stress at work, and so if we can help improve resilience there, we certainly want to think about it. And as you think about the world more broadly, there are constant environmental insults, things like pandemics, which increase all of our stress. So that's how we balance out the ethics between animal experiments, think about things like mice in a swim task, and balancing that out with the impact of ultimately coming up with therapeutic treatments for humans.
Got it. Thank you. Thank you very much.
Yeah.
Any more questions? So, please.
Hello, my name is Abu Bakr. Thank you very much for your inspirational talk. The work you've been through, the hurdles you've overcome, and how you've actually presented it all out in such an amazing way. I think it's quite extraordinary. I have one question regarding, like, you say you're driven by how your work will help your family and possibly people in the developing world. One thing that, from personal experience, I know is that the developing world mental illness is a huge taboo topic. And when you combine that with what we've experienced with COVID in terms of medication and the synergy between the two, how have you taken into consideration that aspect, and how are you looking at the challenges that will come ahead in terms of, like, when you have the drug operational, how would you relay this into the developing world where we have mental illness as a taboo topic and drugs and the medical system?
Yeah, no, I think it's incredibly complicated, right? And I actually spend a lot of my time doing outreach and public engagement as well. Sometimes I think us scientists end up so divorced from the rest of the world that we don't even think through the steps that are necessary, not only to deliver things to people, but to make them understand it and want to take it and bring people in the process, right? So even at the animal stage for me, I spend a lot of time talking to people about research, bringing in folks from all backgrounds and helping them to understand why what we're doing and the strategy is so important. I'm an engineer, so I use design thinking a lot of the time, and that design thinking constrains the choices that I make in the research lab, right? I stopped doing the in silico stuff, although it's like really cool and we still use some of it in the lab, right? I stopped focusing the majority of the lab on that to do this risky thing, right, that was likely to waste a lot of money and unlikely to work, because using design thinking, that was the pathway that made the most sense coming back from the endpoint to where we were now. It made the choices worth taking. So a lot of times I think if there's a future that this works in humans, these are the things I need to be doing right now to prepare humans to think about this. Certainly, COVID-19 has thrown off my impression about the world's ability to deliver, vaccines everywhere, right? And so one of the things that's going to be important is making sure that the tool or the technology in the end is for the world, that it's being developed by the world, and it's not sort of held behind some company where their goal is to ultimately maximize profits off of it. For me, this is really important because, like I said, the majority of my family is sitting in Ghana and West Africa.
Hi, thank you for your inspiring talk. I really enjoyed it, especially that it's personal for you. And I think that the matter of mental illness is also very personal for me. I have had friends and colleagues who had depression, bipolar disorder, or even schizophrenia. And when they are on medicine, they change. They become numb, isolated, very cold. They don't really engage as much as they did. So my question is, did the mice, after having the protein or the worm, I'm not sure, so did they have also a change in behavior towards positive experiences as well as negative experiences or not?
Yeah, so let me answer that two ways because I think it's particularly important, right? The first one is when things go everywhere, they tend to come along with more side effects, right? And I'm not surprised to hear that you've had colleagues that have had depression and schizophrenia and bipolar disorder. I'm not sure what the statistics look like here. In the US, it's like 1 out of 3. Likely in the pandemic, it's probably pushing to like 1 out of 2, right? So it is not uncommon designed for people to have personal experiences with this as well. The challenge with the medications is they likely go the places that you want them to go, but they go everywhere else as well. And so you get side effect profiles with it, and as a clinician, I realized that that's the majority of the time why people stop taking their medication, right? They didn't like the side effects that came along with it as well. So part of the idea of generating this tool was having something where we can send it exactly where we want it. It has to be closed loop. In other words, it has to stay within the appropriate timing of the brain, and that would give us our best chance of not having the side effect profile. As well as we tested the mice so far, we don't see anything. We don't see any problems that come along with it. It normalizes their behavior, but we haven't done an exhaustive series of tests yet. And even if all those tests were fine, we will have only succeeded in curing mouse resilience because, like I said, to the ethics question, a mouse is not a human, right? And so we're taking a series of steps to show with each right model system that we can get the tool to work, but we will ultimately know we are correct when we have put this in humans and shown that we can help people without causing all the problematic side effects that come along with it. But thanks so much for your question.
Thank you.
So just I will take a couple minutes because we have numerous questions online, because there are attendants online, but I'm going to summarize 2 questions. How or what's the way that your research or this discovery, the protein, will be translatable to treatment? How you envision that? How you will deliver this to a human?
Yep. So essentially, we packaged it in proteins because proteins can be put into viruses, right? So the way we put these in the animals' brains is we injected the viruses into their brains. People are working on creating similar versions of exactly the same viruses, except they cross the blood-brain barrier. So you can inject it systemically into a vein, and they will cross the blood-brain barrier. The problem is these viruses don't know where to go yet, right? So they'll go to the whole brain or large sections of the brain. And so there are efforts in the world now to basically create GPS systems for cells in the brain, and that is by sequencing the cells. So I toured your bio facility here in which they're doing DNA sequencing. It turns out cells have unique gene expression profiles, and so if you can learn the unique expression profile for cell type, combine that with the virus, you can express that virus in a specific cell type by putting it in the vein, right? Now, all of a sudden, your closed-loop stimulator is a vaccine, right? And vaccines we know how to develop and deliver.
Thank you.
Yeah.
Another question which came in few questions. How your experience as an engineer or chemical engineering helped you in what you are doing?
Yeah, but so, I mean, as you all can appreciate, right? Like, I'm not married to any given solution whatsoever, right? And I believe there's an incredible value to disciplines, but there's an incredible value to the space between disciplines, too, that people don't explore. So every discipline has its boundary. There's a space where a discipline starts and another one stops. I think there's a whole lot of information in between them. And I'm actually, like, not really terrified to be wrong and to fail. And so I jump around different spaces and use whatever tool is necessary to solve a problem, including sometimes going back to the very beginning again. I think that's engineering 101.
Thank you so much.
Thank you. Thank you all.
Inspiring talk. Thank you. Thank you.